is my pleasure to introduce our speaker today, Dr. Russell Margolis. He is a professor of psychiatry and neurology in the Department of Psychiatry and Behavioral Sciences of the Johns Hopkins University School of Medicine in Baltimore, Maryland. Dr. Margolis received his undergraduate degree from Princeton University and his MD degree from Johns Hopkins University School of Medicine and completed residency training in psychiatry at the Johns Hopkins Hospital. After a fellowship in neurobiology at the National Institutes of Mental Health, where he joined the Johns Hopkins faculty, where his clinical and research interests lie at the interface of psychiatry, neurology, and genetics, with a particular focus on diseases caused by repeat expansions and on schizophrenia. His group, the Laboratory of Genetic Neurobiology, first described the repeat expansion disease Spinocerebellar Ataxia Type 12, which we're going to be learning a bit more about today. Their group discovered the causative mutation and developed a genetic test for the disorder. Dr. Margolis and his colleagues, and in this work now led by Dr. Pan Lee, continue to explore the pathogenesis for SCA12. In addition to his research into repeat expansion diseases and schizophrenia, Dr. Margolis continues to evaluate and treat patients primarily with schizophrenia and to teach students of all levels. And with that, I'll turn the floor over to Dr. Margolis.
Thanks, Celeste. It's a pleasure to be here, even if we're far away from where everyone is, but we're all here together to talk about SCA12. As Celeste mentioned, I'm a psychiatrist, which becomes important at the end of this discussion. But I'm also interested in neurologic disorders with psychiatric manifestations and in genetic disorders in general. This is Johns Hopkins Hospital, or at least part of it. Somewhere right around here is where my current office is. And in 2 months, we get to move to brand new space right in there, which we're very excited about because our current space is crumbling to pieces. We hope there's no fire alarms or floods during the course of this talk. So, let's begin with this. There we go. What I want to talk about today is how we came across SCA12, and then aspects of SCA12, who has it, what are the genetics, what are the clinical features, what do we know about the brain in SCA12, what do we know about the gene that SCA12 affects? What's some of the ongoing research and what kind of treatments are available right now? And hopefully we should have a reasonable amount of time available for questions. So let's start with how we found SCA12, a team effort. So it all starts in about the 1990s. A number of these familial neurodegenerative diseases were shown to be caused by a CAG repeat expansion. We'll talk about what that means a little bit later. 92, 93, 94, 96, and a few others over the next couple years. And so the idea was, it seemed possible that other disorders, like some of these that had been discovered, might also be caused by the same mechanism. So that is not a great leap of logic. Made perfect sense. So as an aside, and this is important for various aspects of understanding SCA12, it might be old news to some of you, newer news to others. It's that the disease, as we will discuss, is caused by a CAG repeat expansion. Understanding that begins with recognizing that DNA is made up of a string of bases, abbreviated A, G, C, and T, that are strung together in a number of different ways. There's about 3 billion of these in the human genome, and how they are organized makes all the difference in how DNA is able to serve as blueprints to manufacture the proteins that do the work of the cell. So I do think it's useful to think of DNA as a blueprint. And from the blueprint, other things are constructed. And of course, it stands to reason if the blueprint has errors in it, that there may be errors in the construction. So in some places in the human DNA, and actually in other species as well, the DNA ends up consisting of a series of repetitions of these bases in triplets. In this case, we're talking about CAG, 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 CAG. And for unknown reasons, in some families, one of these many different repeating regions in the DNA becomes excessively long with too many triplets, which is known as a repeat expansion. And depending on where in the DNA this occurs, the long repeat may cause a disease, and it causes different diseases depending on where in the DNA the long repeat happens. So that's sort of the basic underlying idea that colors much of the work that I'm going to be talking about and the clinical features of the disease of SCA12. And if there's questions about this later, we can come back to it. So we had a two-part strategy that was largely conceptualized by my long-term mentor, Dr. Chris Ross here. One is that we wanted to obtain DNA from patients who had movement disorders of unknown cause. And what we were able to do was set up a collaboration with a very large genetic testing company, Athena Diagnostics, so that they would send us DNA. Of course, it was all to preserve confidentiality. It was just de-identified DNA where we didn't know the person, but Athena did. And the second part of our strategy was to use an assay to detect repeat expansions. And this assay is sometimes simply called the repeat expansion detection assay. And it's just a molecular trick to figure out if somewhere in someone's DNA, there's a CAG repeat that's too long. And this was developed by Dr. Laura Rainham, who was then at the University of Minnesota, is now at University of Florida, but it was implemented very successfully in our laboratory by Dr. Susan Holmes, who is here. And so what we were able to do is take advantage of this strategy. So Dr. Holmes detects a repeat expansion in one of the DNA samples provided by Athena Diagnostics. Athena contacts this individual who agrees to contact us. And from there, we learned about the family, and we were able to set up a big family gathering in one home, which I remember very well because it was kind of crowded. And I spent an afternoon, I obtained consent from everybody, got clinical information, did a blood sample, did a very short neurologic examination of everybody in the family who showed up, probably somewhere about 20 or 25 individuals. Dr. Holmes then took the DNA from all of these family members and found that anybody in the family who seemed to have this disease, which we didn't have a name, we didn't really understand what it was, had a repeat expansion. That's shown in this figure here. These are people who had the disease. Long repeat expansion shown by having lots of these bands going all the way up high. And people without the disease didn't have an expansion. And so we were pretty confident that this repeat expansion likely had something to do with the disease. And then Dr. Holmes used this same assay to locate the specific gene in the DNA where the repeat expansion was located. This was quite a technical achievement that took an awful lot of work. She was very talented, so she could do it in a number of months. Right now, if we had information up to number 4, an undergraduate student could probably do this all in a week. But 25 years ago, it took a very sophisticated scientist many months to be able to accomplish the same thing. That's just the technology so much better, and the human genome is now known and published, and one could use that to make this process go very quickly. We didn't have any of that. So as soon as she was able to find the repeat expansion exactly where it was that seemed to be causing this disease, she could design a simple genetic test so we can figure out if anyone had the mutation as long as we had a blood sample and DNA. So that's the basic discovery. which we accomplished in about the year 2000, I think. And as soon as the genetic test was available, then it could be applied to anybody who had some sort of disease where there was a prominent movement disorder. Our initial family that we found was, because they were located near us, was they're American family, but they were of Swiss-German descent. But what quickly became evident was that most of the cases are from India. Most of the individuals affected with this disease, which by that point we were calling SCA12, because it had features of ataxia, were found, people with the disease were far more likely to be from India than anywhere else, particularly from the Agarwal ethnic group from northern India. And these are three of the most prominent Indian investigators who have really pioneered much of the work in SCA12, particularly clinically. Dr. Srivastava, Dr. Farooq, and Dr. Mukherjee. As genetic tests were tried out in different populations of individuals with movement disorders, a few other cases have emerged around the world. One person in Singapore, two families in Italy, somebody in France, and it is possible that some of these individuals actually had immigrated from India. Multiple families of Han Chinese descent, including, interesting, some people who had mutations both for SCA12, but also for other SCAs, and a large Uyghur family, one of the ethnic populations in China. But relatively speaking, 99% of all cases are from India, and about 90% or so are from individuals from the Agarwal ethnic group. How common is SCA12 in India? Well, a large case series was put together of about over 5,600 or so patients who had some sort of ataxia, meaning gait problems, who were referred for genetic testing. About 27% had some positive tests for one or another of the diseases that could be tested up till the last few years. And it turned out that of the diseases, SCA12 was the most common, that is accounting for about 8.6% of individuals with a diagnosable, genetically diagnosable movement disorder characterized by ataxia. In India, SCA2 is about as common, and the others are far less common. So I did a bit of a calculation. This is one of those napkin sort of calculations. There's an estimated 2.7 people out of 100,000 have SCA in India. That might be an underestimate. There's about 1.4 billion people in India, meaning about a little short of 4,000 SCA cases, meaning that there might be 3,250 people in all of India with SCA12, but I suspect the number is that is not quite right, and it's probably considerably higher based on various underestimates that the Indian investigators believe occur in the health system there. So I would bet it's more in the 10,000 or more range, though it's impossible to know for sure. So it means in the big picture of the world, this is still a rare disease, but it's not exceedingly rare in India and in people who've immigrated from India around the world. I want to talk now about the basic genetics of SCA12. It is autosomal dominant inheritance, meaning that if a parent has the disease, their offspring has a 50% chance of getting the disease. And that is fundamental to thinking about the disorder. The causative mutation, which is defined as having a repeat in the specific part of the DNA that we'll get to in a moment, is how the disease is genetically diagnosed. It turns out it's on chromosome 5 in a gene called PPP2R2B. Normal individuals have generally in the range of 4 to 31 triplets in their DNA, and individuals with 43 to 78 triplets will develop the disease, though there's a little uncertainty for lengths in the upper 30s up to about the upper 40s, because not all such individuals may develop the disease, and maybe having a repeat in the 30s, that is 37, 38, 39 CAG repeats in a row, might be enough to cause the disease. So some uncertainty still. Individuals with 50 or more will develop the disease if they live sufficiently long. We know that a longer repeat is associated with an earlier age of disease onset, which is true of almost all known repeat diseases. And oddly, there are a few rare individuals who have an expansion, who inherited the mutation from both parents, but they don't have a worse case of the disease, which tells us something about the biology, though exactly what remains open for debate. Anticipation has not been observed in SCA12, and anticipation is a phenomenon observed in many other similar diseases where a repeat expands from one generation to the next as it's passed on, and as it gets longer in each subsequent generation, the disease tends to have an earlier onset or a more severe course. So not observed so far in SCA12, though it hasn't been as systematically addressed as possible. It's the kind of thing that would be useful, for example, in genetic counseling of people. Here's the evidence that I don't think has ever been published, showing that as the length of repeat gets longer, and here's the length of repeat from just taking an individual with 50 out to 70 repeats, and here's how old they were when the disease developed. You can see there's a big range, but in general, as the repeat length gets longer, people develop the disease somewhat earlier. Similar association between repeat length and age at onset has been reported in a second study of patients from India. So it's a pretty solid finding and has some importance in providing counseling to people about who want to be tested early in life and would like advice on when they're likely to develop symptoms, though it will be a very approximate guess. So let's talk about the basic clinical features of SCA12. So as I mentioned, the onset's been variable. There's been individuals who had an onset as early as age 8 to later than age 60, though it's typically middle age, late 30s to early 50s. Though interestingly, again, this hasn't been as systematically studied as one would have wished. The initial clinical presentation, the symptoms that people first notice, are tremor, usually in the arm, sometimes in the head. An action tremor means that it's a tremor that's more obvious when someone's, say, reaching out or holding a cup of coffee or something, rather than just sitting at rest, which is very different than Parkinson's disease, which tends to be the other way around. People may have gait ataxia, that is unstable gait, fairly early, and sometimes will have some slurring or dysarthric speech early. Over time, the tremor tends to become more prominent and to involve more body parts. The gait imbalance becomes more severe, and other clinical features tend to emerge over time, but variably in different people. Some people have facial myokemia, which just means kind of like wiggling of the facial muscles. Eye movement abnormalities, where if you ask someone to track a finger moving back and forth, they'll have some difficulty or be slow. Dystonia, which means a sustained twisting like this, or can be in a neck like that. I don't know how well I could be seen. Hyperreflexia, which just means that reflexes are overly strong, which can cause trouble when people are walking or doing other movements. Dysmetria means that people have a hard time reaching a target. So if you ask someone to point from their own nose to the examiner's finger, they'll tend to have trouble getting the point exactly right. And that's Same thing can be seen in feet, which you can imagine would make walking somewhat more difficult. Some individuals develop mild symptoms of Parkinsonism, which is characterized by a somewhat hunched posture and a slow shuffling gait and stiffness. And a few individuals seem to have what's called subclinical peripheral neuropathy, meaning that they either have difficulties with sensation, but very subtle. They don't feel things as well. And some individuals may have some problems with pain, but that seems to be relatively rare. So that's the most common motor features. There are late features that seem to suggest a more general involvement of the nervous system. One is primitive reflexes of the Babinski sign, which are just suggesting that some of the pathways from the brain down to the feet or to the hands are in some way being disrupted. Dysphagia is swallowing difficulties, which obviously can have importance. And autonomic dysfunction is often means blood pressure regulation problems, that the nervous system that partially controls blood pressure doesn't adjust very well when a person stands up. So they might feel faint or even actually faint, which of course has consequences. We will talk more about the neuropsychiatric features that can emerge: anxiety, depression, cognitive difficulties, and late cognitive difficulties that are far enough along to be defined as dementia. There are a few major themes compared to other ataxias, the other SCAs, that are important in understanding SCA12. One is the disease tends to be later than most of the other diseases caused by repeat expansions. The onset is later. It is generally milder, unfortunately milder, than many of the other disorders. The progression as it gets over time is slower than with many of the other similar disorders. And there's a great deal of variability in which features will emerge and will be most problematic for an individual with SCA12. As I mentioned before, I'm a psychiatrist, and I'm particularly interested in the psychiatric manifestations of SCA12, which turn out are quite common. This is one of the few studies that was published that did a fairly systematic look at a case series of 21 patients who were fairly typical. They had a mean repeat length of about 55 and a mean age of onset of 51. So this is an average or somewhat older than average patient population. And they found a series of psychiatric manifestations: agitation or aggression, irritability, lability, apathy, anxiety and depression, eating and sleeping difficulties, disinhibition, and even delusions and hallucinations or euphoria. What I would caution, however, is that if you ask 20 people in the general population these same set of questions, the answers would not be zero. So this is, it's not quite as severe as it looks without a comparison group, but it's not trivial. And we'll come back to this because these are often treatable aspects of the disease that we don't want to miss. There are a few less common clinical presentations that have fooled clinicians over time, and there are scattered reports throughout the literature of this sort of thing. One is that some people have chorea, which is quick little jerking motions, that is more commonly seen in disorders like Huntington's disease. Some people have a spasmodic dysphonia, which is, rather than just a slurred speech, having a kind of a halting, broken speech with a strained voice. And some people have a presentation that is similar to a disorder known as progressive supranuclear palsy, which is characterized by a different sort of walking problem and most characteristically an inability to look up or down, even though patients can look sideways. And so I think the big picture here is that SCA12 can look like other disorders. And so one, in viewing one's own clinical course, one shouldn't be too surprised if unusual symptoms emerged, and clinicians shouldn't be surprised to see these different variations in the disease presentation. How about the functional impairment of individuals with SCA12? The big point is that it tends to evolve slowly over years to decades. There are plenty of examples of people who are functioning quite well in life, continuing to work, continue to be involved in their families for years and years after they begin to develop clinical symptoms. And I take that's actually very good news that people need to remember. But there is some functional impairment that occurs over time. One can imagine that if you have problems with tremor and coordination, you're going to have problems with dexterity, that you'll have problems with walking, and that overall your ability to function may decrease some. In practical terms, that may mean difficulty with writing, handling liquids like pouring something from one container to another, being able to brush your teeth or do dental flossing, dressing because buttons are difficult to do, using a phone or computer because of the fine finger motions, and cognitive difficulties, which we'll come back to. So there is functional impairment, not nearly as severe as in other related disorders, but enough that families and individuals will need to pay attention to it and plan for the future. What about the brain? What's emerged is that there are brain findings that are not just limited to the cerebellum, but involve the cerebral cortex. So here's an MRI scan of an older man who has a fairly short repeat length. And what is pretty obvious is this is the guy's cerebellum. If you took a cross section right through his nose in the middle. And so the cerebellum should fill out this whole space here, just about, and it shrunk. And the same thing, not quite as clear, but the gray stuff here is cerebral cortex, and the white stuff is cerebral spinal fluid. And this is the fluid fill cavity in the middle of the brain, which is in this individual's a bit large, and there's little bit too much of this fluid outside his brain, meaning that his brain, the cerebral cortex of his brain, has shrunk some. And that's very typical of findings. And one of the most important factors of this facts about SCA12, it's not limited to the cerebellum. It affects other brain regions. And it is because of that clinical symptoms emerge that are not just those of coordination, but also involve a more prominent difficulties with thinking and memory and the like, but only over time. Here's an interesting example of a man who had onset of disease at age 40 and trouble walking at age 44, and had quite extensive symptoms by 10 years after disease diagnosis, but without major cognitive deficits. And he's still working as a physician. And you can see here again is the shrunken cerebellum in this part of the brain, and also pretty major shrinkage of a cerebral cortex. But while it led to symptoms, he was still able to function. And that's important to remember. I want to talk now about one particular individual. She was the first person to come to attention to us and led to our discovery of the disease. And so we knew a little bit more about her. By age 38, she had tremor, which moved to include her head by the time she was in her 40s. Walking became more of a problem by the time she was in her 50s. And by her early 60s, she was quite impaired with psychiatric problems, difficulty with walking, and attending to activities of daily living. And we examined her at that point, and she had dementia This is a rating scale of a very simple bedside scale of cognition, the mini mental state examination. Normal score is 25 and higher, and so she was markedly impaired. This, you might see the same sort of score in somebody with moderate Alzheimer's disease. She had tremor. She had all the symptoms we've talked about just about before, and she became debilitated and ultimately died from pneumonia after she was quite ill. And very generously, she and her family donated her brain, which is important because it's the only SCA12 brain that's ever come to scientific attention, probably because of restrictions on tissue donation in India. I'm told there are one or two other brain in pathology labs in India, but they have not been studied. And I credit much of the work I'm about to show you to my late colleague, Dr. Elizabeth O'Hearn, who helped define the clinical syndrome of SCA12 and was an expert with neuroanatomy. So she took this picture of this brain, and it shows the shrinkage. These are the gyri and sulci, the valleys and peaks of the brain, and there's just too much space scattered throughout much of the brain here. And microscopically, if you look at the cerebellum, a normal cerebellum looks something like this. And these are these great big cells here, these pink ones. They're called Purkinje cells, and they are the main cells that send output messages from the cerebellum. And they're evenly spaced, and you miss a few here because of the way the section is cut, but they're there. But if you look at an individual, this one individual with SCA12, many of the Purkinje cells are gone. And that explains many of the symptoms of the illness. And what Dr. O'Hearn showed with these very beautifully stained images, microscopic images, was that there are these little inclusions that she detected in some of the cells in this individual's brain that stained with some particular stains and not with others, and most importantly, did not stain with an antibody that is detecting long stretches of polyglutamine. And we'll come back to why that's important later, but it means that SCA12 is different than other CAG repeat disorders, where the mutant gene encodes a long stretch of the amino acid glutamine in protein, which clumps up in cells. And we don't see that in SCA12, meaning that the disease is probably different in how, in its pathogenesis, how the mutation causes neurologic damage than in many of the other diseases. So how does the SCA12 mutation cause disease? Well, the mutation is located in the gene PPP2R2B, which doesn't have much meaning in and of itself. It's located on chromosome 5 out of the 23 chromosomes in the human genome. Everyone has 46, one X, one Y, and 22, 2 copies of the other 22. So this is on chromosome 5. This gene is the blueprint that encodes 2 almost identical but not quite identical proteins that are called B beta 1 and B beta 2. And it's these proteins help regulate other proteins. And the current conception that we and others have is that the mutation, the SCA12 mutation in the gene PPP2R2B, where there's too long a stretch of CAGs in the middle of this gene, may affect the gene in a couple different ways. One is that it caused too much of the gene to be made into proteins. I think too much to be made into B beta 1. Another is that it changes the way the gene is organized, because genes tend to be spliced in different ways. Genes come in, consist of a number of different parts known as exons, and the exons can be spliced together in different ways, and the splicing may be abnormal with the presence of the repeat. And the repeat may directly encode a protein that is toxic, which we will come back to in a minute. In this case, it's not glutamines, but too many of the amino acid serines. So there's evidence for all of these functions. Exactly which one is the most important is not clear, and that makes a big difference because it will change how therapy is developed. I throw this out just to let you know that I greatly oversimplified everything, that this gene PPP2R2B is really complicated. This is a standard kind of gene map. The 1 through 16 are the different exons that are spliced together to form the part of the gene that codes protein, but they're spliced in all sorts of different ways. The repeat occurs right here, somewhere in exon 7, but even then there's different versions of exon 7, and which part of the gene is assembled as it's made into the intermediate product RNA makes a difference in what protein is made, et cetera, et cetera. And puzzling all this out has taken years, and I'm sure that we don't have it right yet. On the other hand, then we look at what does the protein that this gene code for do, and it turns out that it is this complicated regulatory protein. It's 3 different proteins work together to generate an enzyme that regulates other proteins. And the PPP2R2B gene encodes part protein B, but there's lots of different protein Bs, not only from PPP2R2B, but from lots of other related genes. And so there's all sorts of versions of this 3-part protein, which varies in what it does based on which part of the protein is attached to this scaffolding section A and this action-oriented center C. B is the region that tells this 3-part protein where to go. And there's about 92 different versions of this. And we're interested in 2 of the 92 versions. So it gets complicated about what's happening to this protein when the mutation causes more or less of this regulatory unit to be made. Again, that would be a long story. I just wanted to give you a taste of what the kind of complications are that we face in understanding this disease. So how can we study SCA12? One is to look at brain tissue. That would be really important. I'd like to have more. The other ways are to develop mouse models that have the mutation. So we fortunately have Dr. Pan Lee, who's my junior colleague, who is rapidly advancing in the academic world, who's an expert genetic engineer. So she has developed both mouse models of SCA12 and human stem cells that have the SCA12 mutation. And it's a long and involved process, but she's an expert in it, and it gives us very valuable tools. We also are fortunate to collaborate with Dr. Stefan Strack from the University of Iowa, who happens to be an expert in PPP2R2B and the genes that it's connected to. And he has a variety of scientific tools that he also is applying to this project. So what is the effect of, if you make too much of PPP2R2B because of mutation, it can lead to cell toxicity. I just give you one little experiment where you put too much of this gene in a cell, and the cells, this is a normal cell, these are cells with too much of the PPP2R2B expressed in it, and cells die in culture. This is a control of something that's known to be toxic. The mouse with the mutation has 2 major changes. Dr. Lee has detected. One is it changes the splicing of PPP2R2B, as I mentioned before, and it changes the genes that are expressed within the brain. And again, the details aren't important. It's just the fact that with these model systems, we're able to begin to understand a little bit more about how the disease arises from the mutation, but there's a whole lot more work to be done. And in mutant mice, as I mentioned, sometimes the repeat might encode too many amino acid serines in a particular protein, and that's toxic. And that's what these are other cell models that Dr. Lee has developed. And so it's another way that the mutations could cause disease, though exactly which mechanism is most important, we don't know. This remains very speculative. So what can be done clinically about SCA12? There's no medicine or treatment that can stop the progress of the disease yet. Therefore, all treatments are designed to control symptoms. General care can make a big difference, maintaining good health, proper exercise and weight. And then rehabilitation and maintenance of function can be assisted. For example, physical therapy. An individual can develop practices to improve balance and obtain devices to assist with walking. Not that this will cure the illness, but it can help slow some of the functional impairment. The same thing can be done with occupational therapy, where the focus is on use of hands and adapting a home environment so an individual is less likely to fall and can manage their activities of daily living more readily. And speech and language experts can help individuals maintain clarity of speech, and even more importantly, if they develop swallowing troubles, develop adaptations that can help prevent choking. There are no treatments that are specific for SCA12, but there's some other treatments that have been proposed for other similar disorders that may be of help. A lot of talk about antioxidant medicines, though there's very little evidence that they're effective. There are some evidence for abnormal movements. Tremor can often be treated with medicines like propranolol or with the medicine primidone. Nerve-related pain might be treated with gabapentin and other related medicines. Muscle spasticity can be treated with baclofen. Erectile dysfunction, which affects some people, can be treated with sildenafil and other medicines. There is some work on non-invasive brain stimulation, which is you don't need surgery, you just have some focal magnetic forces applied to the scalp. Unclear how much these various forms of non-invasive brain stimulation are of help. Also work underway in a number of the disorders, less so in SCA12. And there has been some efforts to surgically control tremor through deep brain stimulation, such as implanted electrodes, or with ultrasound-induced lesions, actually damaging the thalamus to try and prevent tremor. Whether these are effective and worth the side effects, not clear at all. The psychiatric manifestations are important and often treatable, and this is something that as a psychiatrist, I want to emphasize. One is general education for the family and the patient about what to expect with the disease, and sometimes actual therapy or counseling for the psychological effects of having a neurodegenerative disease can be valuable. Depression is relatively common, as we've seen in SCA12 and in a number of other similar brain disorders, and it's easy just to dismiss depression as someone being upset because they have an illness, but in fact, it can be a manifestation of the circuitry in the brain that's injured and not a psychological factor. And individuals can respond to typical antidepressants, at least based on other similar diseases, as there's very little work on SCA12 itself, starting usually with lower dosages. Some individuals may benefit from brain stimulation techniques for depression, like transcranial magnetic stimulation, and electroconvulsive therapy if the depression is severe. Treatment can make a big difference. Similar with anxiety, which can be treated with antidepressants probably most effectively, as well as psychotherapy and support. Sometimes short-term use of benzodiazepines, medicine like Valium or Ativan, can be helpful in the short term, but they can cause problems with balance and can lead to longer-term problems. And the cognitive impairment in dementia, fortunately, tends to become problematic only late in the course of the illness. And the recognition that cognitive impairment is occurring is important so that families and individuals alter expectations and can make life adaptations as possible, like making sure that family is there to provide support, making sure that financial arrangements are taken care of. Irritability is, as we see, can be common. And the first step is always to limit the kind of stimuli that might trigger irritability. So that may involve changing family interactions or other things that upset a person. Irritability may respond to antidepressants or to mood stabilizing medicines. Occasionally, low dose of antipsychotic medicines can be helpful. Apathy is difficult to treat itself, so it's important to make sure it's not depression. If someone's not interested in doing something, that it is not part of a feature of depression, which can be tricky to distinguish sometimes. Here, the goal is behavioral guidance for the individual, gentle, structured expectations, and avoiding major conflicts, which just make matters worse. You can't yell at somebody to get them to do things if they just have no initiative. Occasionally, people have hallucinations and delusions. And here, the first thing is to make sure there's no medical cause like an infection. And these individuals may benefit from antipsychotics, though again, keeping the dose low. The future really depends on where the research will go. We clearly want to stop the disease before it causes damage. It is possible that medicines will be discovered for other neurodegenerative disorders that will turn out to be helpful for SCA12. That has not yet happened. There may be specific treatments for SCA12 that might emerge from the kind of research that Dr. Lee and others are doing, such as genetically changing the DNA or blocking the amount of excess protein or other gene products that are causing problems. Too early to know if this will be helpful or viable, but I think something well worth pursuing. I think I'll end with that and leave us with some time for questions. I should have left a few more minutes, but oh well, this is what we got.
Would you be able to stop your screen share? Fantastic. And we already have some questions in the Q&A window. Just a reminder, if you do have questions, to put them in the Q&A window. We aren't able to unmute anyone if you raise your hand, so we ask that you type them in there. We're going to get through as many as possible. So first, we have an attendee who is asking, what is a good time to get genetic testing done? She shares that she has a mother who has SCA12 and is in advanced stages. She has the gene. She's not sure if her daughters have it, though, who are young adults. Could you comment on this?
Yeah, I think that's very much an individual decision, especially young adults have the right to get pre-symptomatic genetic testing. That usually should involve some genetic counseling. That's been well established for more common diseases like in this family, less so with SCA12. But some individuals want to know and some don't. And each person has to make their own decision. And that's where the advantage of counseling is, because there are all sorts of possibilities ranging from illegal but possible discrimination to just psychologically. Do you want to know or do you not want to know what the future holds? And each person is different.
Very good to know. We have another attendee who asks, does the number of repeat expansions tell you anything about severity of symptoms, or is it mainly about age onset of symptoms?
I think the evidence about that is mixed. clearly age of onset correlates, but not tightly. And the literature about the severity and nature of symptoms is all over the place. So I don't think I can give a really good answer. My hunch is that it probably does, but maybe more at the extreme ends, the very longest and the very shortest repeats, but with lots of exceptions.
Well, it's good to know that there is that uncertainty. We have another question from an attendee who asks any thoughts or information about why SCA12, unlike other genetic forms of SCA, typically has later onset. What's known about that?
I think one of the reasons is that it's not a polyglutamine disease. Most of the other CAG repeat expansion diseases that are called SCAs um, are caused by the gene product, the gene coding for a long stretch of polyglutamine, which we know is toxic. SCA12 looks like it's more complicated and more subtle, but that still remains a bit of a mystery and a clue of some sort for sorting it out.
Very good to know. We have another attendee who shares that they have the SCA12 gene. Uh, his mother had SCA12. Do you always develop symptoms if you have the gene? And is there anything you can do as preventative measures before you have symptoms?
Generally, if you have a mutation, the gene that is the mutation in the general age range, the length that causes disease, you will eventually get the disease, though exactly when is pretty hard to predict. When a parent got it is probably one good index, but not perfect. Um, there is no solution to preventing its onset, but it certainly makes sense to be healthy, be in good condition, make sure that you're is good, and it helps to be able to make some plans for the future. Not huge plans, but little things about, do you want to make sure that you have um, supports in your shower to hang on to, and or do you want to consider in 10 or 15 years needing to move to a place with no stairs? Things like that that aren't emergency things, but are worth considering.
Very good to know. We have another question from an attendee who shares that they live in northern India. They have a biallelic mutation of SCA12. They come from a large family with multiple members impacted. They're interested to know what ways do you quantify progression of SCA12?
Yeah,
How do you do that in clinic?
yeah. There's a number of different ways. There are various rating scales for gait. There are rating scales for cognition. There are rating scales for tremor, and kind of a rating scale overall from uh, binding all the different symptoms into one rating scale. And fortunately, the clinicians in northern India, the neurologists, at least the ones I know of, are quite expert in this and can help provide very specific answers to that.
And then we've had a few people ask a very similar question. Is epilepsy a symptom of SCA12?
It doesn't seem to be, though I would caution that any neurodegenerative condition that affects the brain somewhat diffusely, like SCA12 does, probably increases the risk somewhat, but that's pure speculation.
Mm, very good to know. And then we have a follow-up question because we were talking a little bit earlier about the genetics and how this is an inherited form of ataxia. Is there a way to um, prevent transmission of the gene from parent to child? What kinds of reproductive health options are available for folks who have SCA12?
I can speak theoretically because I
Yep.
don't know what's practically available. And I can speak from the Huntington's disease literature that one can do in vitro fertilization where embryos are created in a dish and tested, and only embryos that don't have the mutation are then implanted. That, of course, is a really expensive and invasive procedure um, and not available everywhere. There are more extreme would be individuals could be, it is conceivable, though I don't know if it's done, that individuals can be uh, tested during pregnancy to see if the fetus has the mutation, and I suppose then a selective abortion could be obtained. that again, I don't know that's ever been done with SCA12, given that it's such a late onset disease, but it is conceivable. So those are the main strategies.
Thanks for walking us through those options that could potentially be available. We have another attendee who asks, you mentioned some of the symptomatic treatments that are available. Are there any drugs available to help improve balance and walking?
Not really, to be quite blunt about it. At least none that I know of. I think there it is physical exercise, balance, practice with balance, working with a physical therapist is necessary, but there are not drugs that will make a big difference that I know of.
That actually leads well into the next question that an attendee asked. They're wondering if you could reiterate some of the proactive measures that people could take, such as exercise.
Yeah.
Could you speak about that a little bit again?
Sure. Most of this is pure common sense, and um, amount of actual evidence is small, but being in good physical shape is better than not being in good physical shape. If one being overweight will impair balance some. Being physically adept in general is a good thing, and maintaining that even while ill, even as the illness begins, is going to be of value. There's nothing perfect about any of these rehabilitative measures, and much of it is pure common sense.
All right, I think we have time for two more questions. The first being an attendee is asked, is general intelligence impacted by SCA12?
Eventually, probably in many people, yes, but later in the course of the illness, and not necessarily so severely as to cause major impact on life, but it's quite variable.
And then our last question, we have an attendee who asks, from the brain histopathology of SCA12, are there any deposits or plaques? Is this causing aggregates in the brain?
Maybe, but not the kind seen in Alzheimer's disease, not the kind seen in Parkinson's disease, and not the kind seen in Huntington's disease. There is some only one brain's become available, and that brain from a very severely affected individual showed a few odd inclusions. It might be a clue, but might turn out to be a red herring. It is possible that our animal models will help with this, but having access to more brain tissue will make a big difference, and that may help understand the disease much better.
Well, thank you so much for your fantastic presentation, Dr. Margolis, and sharing your expertise with us. We really appreciate it.
My pleasure. My best to all of those who have SCA12 and their family members, and it's a difficult and challenging illness, but one for which there's hope, and people live long and high quality lives despite having the illness.
And thanks to everyone who was here on this session. I hope you found something valuable and learned something new about SCA12. And wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take care, everyone.